Hello there, my name is Taysir Arajabi, and I am a student at the University of Kansas. Today I'm going to be giving you a presentation about the kinetic behaviors of apigenin in chamomile tea when paired with the enzyme glycogen phosphorylase. Now, glycogen phosphorylase is an enzyme commonly found in the body that is responsible for releasing glucose in the form of glucose 1-phosphate from the storage form glycogen. There are two types of glycogen phosphorylase, namely glycogen phosphorylase A, the active form, and glycogen phosphorylase B, the less active form. In this case, we are going to be studying glycogen phosphorylase B because it is much easier to realize activator behavior from a less active enzyme. The effectors that I've chosen for this study are chamomile tea itself and apigenin, the active ingredient in chamomile tea. This is because people who drink chamomile tea often experience feelings of drowsiness and low energy, and the flavonoid molecule responsible for this, apigenin, has a variety of interactions with many parts of the body. So I believe that it will have a binding function with glycogen phosphorylase and eventually produce an inhibitory effect on the enzyme itself. Now, the way to prove this is that we're going to be running the experiment in reverse. If you look in part A of the background, the reaction of glycogen phosphorylase is that it uses inorganic phosphate to free glucose from glycogen itself. But when running the reverse part, we are going to be using glucose 1-phosphate as our substrate and quantifying the amount of inorganic phosphate that is produced, namely through the absorbance values. Absorbance spectrometry can produce a numerical value for how much phosphate is produced here. In our first results, in the top right of our experiment here, we started running with a known activator of glycogen phosphorylase B, specifically AMP. When energy levels are low, usually there's an abundance of AMP in the body, and this was proven by the decrease in Km and the increase in Vmax at higher concentrations of AMP. Moving on to the main results of this study is that with high concentrations of chamomile tea, we realized that there was an increase in Km and a decrease in the Vmax. When the Km increases, the affinity of the enzyme for its substrate decreases, whereas when the Vmax increases, the rate of reaction of the enzyme also increases. Because the Vmax decreased and the Km increased, these are hallmark indicators of a mixed inhibitor. Whereas we had some stranger results with apigenin in which the Km increased by a significant amount, but so did the Vmax. There is no type of inhibitor that can increase the Vmax, so ultimately our results for apigenin were inconclusive. Because of this, our future directions would be to run more trials to really confirm that this data is accurate, as there may have been some erroneous points or some statistical error that may go into this experiment. Other avenues that we could pursue would be 3D modeling of the enzyme itself using a software such as SwissDoc to see if there's an actual binding site for apigenin as a modulator of this enzyme. Furthermore, another experimental trial that we would like to try is with a known flavonoid inhibitor of glycogen phosphorylase B. One example is flavoperidol. This type of enzyme can bind to glycogen phosphorylase B and decrease its activity. These research results would not be possible without my lab partner, Clayton Brockard, and I would also like to thank our mentor from this lab, Dr. Roberto de Guzman.